Hello, it's Joni Rotten. I wanted to read this article. I don't know why, but let's read it. This is from April 16th of 2018, and it says Epstein Barr virus linked to 7 serious diseases by the Cincinnati Children's Hospital Center. There you can see the 2 viruses. I'm probably going to chop up a lot of this, but we're going to try it anyway. A far-reaching study conducted by scientists at Cincinnati Children's reports that the Epstein-Barr virus, or EBV, best known for causing mononucleosis, also increases the risk for some people developing 7 other major diseases. Now, you can get sick with this virus and not develop mono. Not everybody. Remember, Epstein-Barr virus lies within 95% of the population. So it's there waiting for something to trigger it. In some cases, people get mononucleosis. There are these 7 diseases that are also linked are systematic lupus, erythematosus, or SLE, multiple sclerosis, MS, rheumatoid arthritis, MA, juvenile idiopathic arthritis, or JIA, inflammatory bowel disease, IBD, celiac disease, and type 1 diabetes. Combined, these 7 diseases affect nearly 8 million people in the United States. Study results published April 12th in the journal Scientists, and then it names a few. John Harley, he's an MD, PhD, director of the Center for Autoimmune Genomics and Etology, CAG is the best way to put it, at Cincinnati Children's, and a faculty member of the Cincinnati VA Medical Center. Leah Kotian, she's a PhD, and immunobiologist expert with CAGE, and another guy named Matthew Wurich. He's got a PhD, and he's a computational biologist with the center. The critical contributions were provided by a few other people that have letters behind their name. The study shows that a protein produced by the Epstein-Barr virus called EBNA2 binds to multiple locations along the human genome that are associated with these 7 diseases. Overall, the study sheds new light on how environmental factors such as viral or bacterial infections, a poor diet, pollution, and other hazardous exposures can interact with the human genetic blueprint and have disease-influencing consequences. Quote, now using genomics methods that were not available 10 years ago. It appears that components made by the virus interact with the human DNA in the places where the genetic risk of disease is increased, Harley says, and not just for lupus, but for all these other diseases too. The full impact of the study could take years to explore. Here are some of the initial implications. So we have new concerns about the kissing disease. EBV, Epstein-Barr virus, is a strikingly common virus. In the United States and other developed nations, more than 90% of the population becomes infected by age 20. In less developed nations, 90% of the people become infected by age 2. Once infected, the virus remains in people for their entire lives. Mononucleosis, which causes weeks of extreme fatigue, is the most common illness caused by EBV. Mono was nicknamed the kissing disease years ago because the virus spreads primarily via contact with saliva. Over the years, scientists have linked EBV to a few other rare conditions, including certain cancers of the lymphatic system. Harley, who has devoted much of his career to studying lupus, found possible connections between lupus and EBV years ago. That work includes proposing mechanisms that the immune system uses in response to the virus that leads to lupus, and showing that children with lupus almost always are infected with EBV. Today's studies adds weight to those lupus findings and adds six more well-known diseases to the list. This discovery is probably fundamental enough that it will spur many other scientists around the world to reconsider this virus in these disorders, Harley says. As a consequence, and assuming that others can replicate our findings, that could lead to therapies, ways of prevention, and ways of anticipating disease that do not now exist. So far, no vaccine exists that will prevent EBV infection, although let me say that they are working on it. I think we've come up with and in fact, they are working on it. This article being a little bit older, you can find newer stuff. They've just pulled it off the market for a minute because somebody had some side effects. I think I think we've come up with a really strong rationale for encouraging people to come up with more of an effort. Catan says. Yet, some EBV vaccines are under development. I think the study might well encourage them to push forward faster and with rededicated effort. How does EBV hijack our immune system? When viral and bacterial infections strike, our bodies respond by commanding B cells within our immune systems to crank out antibodies to battle the invaders. However, when EBV infections occur, something unusual happens. The EBV virus invades the B cells, reprograms them, and takes over control of their function. The Cincinnati Children's Research Team has discovered a new clue about how the virus does this, a process that involves tiny proteins called transcription factors. Our bodies have about 1,600 known transcription factors at work within our genome. Each cell uses a subset of these to become what they are and to respond to their environment. And these proteins consistently move along the strands of our DNA, turning specific genes on and off to make sure cells function as expected. However, add. When the transcription factor changes what they do, the normal functions of the cell can also change, and that can lead to disease. The Cincinnati Children's Team suspects that the EBNA2 transcription factor from Epstein Barr virus is helping change how infected B cells operate and how the body responds to those infected cells. The new paper shows that seven seemingly unrelated disease states actually share a common set of abnormal transcript factors, each affected by the EBNA2 protein from the Epstein-Barr virus. When these EBNA2 related clusters of transcription factors attach themselves to one portion of the genetic code, the risk of lupus appears to rise. When those same transcription factors land in another part of the code, the risk of multiple sclerosis appears to arise, and so on. Normally, we think of transcription factors that regulate human gene expression as being human. Um, Katyan says. But in this case, when the virus infects cells, the virus makes its own transcription factors, and those sit on the human genome at lupus risk variants and at the variants for other diseases. And that's what we suspect is increasing risk for the disease. New leads emerge for improving treatment. It remains unclear how many cases of the seven diseases listed in the study can be traced to prior EBV infection. More genomic analysis involving many more patients with these diseases will be required to make reliable estimates. The impact of the virus is likely to vary across the diseases, Harley says. In lupus and MS, for example, the virus could account for a large percentage of those cases. We do not have a sense of the proportion to which the virus could be important in the other EB and A2 associated diseases. However, the breakthrough identification of specific transcription factors connected to EBV infections opens new lines of study that could accelerate efforts to find cures. The same cast of characters is a villain in multiple immune-related diseases. They're playing where Weirach says. W says. They're playing that role through different ways and doing it in different places in your genome. And it's the same sinister characters. So, if we could develop therapies to stop them from doing this, then it would help multiple diseases. A number of compounds, some experimental, some approved as medications for other conditions, already are known to be capable of blocking some of the high risk transcription factors listed in the paper, he says. And teams at Cincinnati Children's have begun deeper studies of some of these compounds. Findings go far, far beyond EBV. While the EBV related findings involve more than 60 human proteins linked to 7 diseases, the Cincinnati Children's Research Team already has taken a huge next step. They applied the same analytic techniques to tease out connections between all 1,600 known transcription factors and the known gene variants associated with more than 200 diseases. The results of that mass cross analysis also appear in today's study. Intriguing associations were documented involving 94 conditions. Our study has uncovered potential leads for many other diseases, including breast cancer, Hurley says. We cannot possibly follow up on all of these, but we are hoping that other scientists will. After devoting decades of research to hunting down the causes of lupus, Harley says this study represents the most important discovery of his career. I've been a co-author in almost 500 papers. This is more important than all of the rest put together. It is a capstone to a career in medical research, he says. The software behind discoveries to be made public. So, detecting and tracking the activities of these transcription factors took years of work involving dozens of laboratory and computational experts. The project required gathering massive sets of genetic data, then analyzing every genetic change affecting the activity of the virus. Doing this required creating 2 new algorithms called RELI and MARIO, or Rely and Mario, which were developed in Cincinnati Children's by W and his colleagues. Both software tools and a related website will be made publicly available. Quote, we are going to great lengths to not only make the computer code available, but all of the data and all of the results, he says. We think it's an interesting approach that could have implications for many diseases. So we're contacting experts on the various diseases and sharing the results and seeing if they want to collaborate to follow up on them. If you want more information, you can contact if you want to be a part of Nature Genetics. Found that to be an interesting article. I hope you did too. We're going to be finding out, I think, that a lot of things are going to be linked to these viruses.